All right, let's jump right in. We're about to explore one of the absolute biggest questions in science today. What is going wrong inside our brains as we get older? But here's the twist. We're going to connect that mystery to the world of artificial intelligence, because it turns out the mind of a machine and a brain in distress, well, they might have a whole lot more in common than you think. To really get why this is so important, just look at this number. $345 billion. That's how much Alzheimer's disease costs in the United States every single year. So yeah, this is way more than a scientific puzzle. It's a massive, heartbreaking crisis that affects millions and millions of families.
So what is actually causing all of this devastation? That's really the core question we're digging into here. Right now, scientists are locked in a huge debate over what's fundamentally happening in diseases like Alzheimer's, Parkinson's, and ALS. And honestly, the answer could change everything.
We're talking about millions of people affected by these neurodegenerative diseases. And for decades, the best minds in science have been trying to figure out the root cause. The whole debate really boils down to two completely different ideas about how the damage spreads. Is it something contagious spreading from cell to cell, or is it the whole system just breaking down?
Okay, so let's start with the first big hypothesis. The easiest way to think about it is as the contagion theory. This idea suggests that these diseases aren't just popping up randomly. No, it suggests they are actively, physically spreading from one brain cell to the next.
Now, the official name for this is the synaptic spread hypothesis. The core idea is pretty straightforward. You have these toxic, misshapen proteins, and they are the villains of the story. They don't just sit in one place, they actually travel across the brain's incredible network, spreading from one neuron to the next through their connections called synapses. You can see how it works here. It's like a cascade. It all kicks off in cell 1, where a single protein folds the wrong way. This one rogue protein then acts like a bad seed, causing other healthy proteins around it to misfold too. Once enough of these toxic clumps get together, the cell basically spits them out, and they get picked up by the next neuron in line, cell 2, and the whole destructive process starts all over again. It's this slow, methodical, relentless spread.
So, here's the crucial part. For this contagion to work, it needs a highway to travel on, and that highway is the brain's own communication network. So to really get the theory, we need a super quick refresher on how our neurons actually talk to each other.
All right, so neurons use electrical signals to chat. Sometimes one neuron will send what's called an excitatory signal over to its neighbor. You can just think of this as a go signal. It gives the next cell a little nudge, telling it, hey, get ready to fire. But, it can't be all go, go, go. So neurons also send inhibitory signals. These are the stop signals. They do the exact opposite. They tell the receiving cell, nope, stay quiet for now. And this balance between the go and stop signals is absolutely critical for a healthy brain to work.
And this is where you really see the brain's insane computing power. Any single neuron is getting thousands of these go and stop signals all at once from all its neighbors. It's constantly adding them all up. And if the go signals win out by enough, boom, it fires its own signal. This is the very network, the very system that the synaptic spread theory says the disease hijacks to spread.
Okay, so let's shift gears and look at the competing idea, which paints a totally different picture. This one is called the weakest link theory. And instead of a contagion spreading through a perfectly healthy network, this theory says the network itself has vulnerable spots from the very beginning.
The formal name here is the selective vulnerability hypothesis, and it argues that the disease isn't really spreading at all. Instead, it says that certain types of neurons are just naturally weaker or more stressed out than others. It only looks like it's spreading because these vulnerable cells are failing in a predictable order, kind of like a string of cheap holiday lights going out one by one.
So, you're probably wondering, what makes a neuron a weak link? Well, scientists have a few ideas. Some neurons might just have to fire way more often, putting them under constant stress. Or maybe they're just more easily damaged by things called free radicals. It could even come down to a tiny imbalance of metals, or their unique shape, or even the specific genes they're using.
So now we have two totally different explanations for the exact same tragic outcome. One theory says the connections are the problem, the other says it's the components themselves. So how do we figure out who's right? Well, it turns out both of these ideas might actually be pointing to something much deeper, a more fundamental principle that's at work in the background.
Let's just put them side by side. Synaptic spread is that contagion model. Think of it like a domino rally. One piece falls, and it's guaranteed to topple the next one in line.
The network connections are everything. On the other hand, selective vulnerability is the weakest link model. This is more like a city's power grid failing during a heat wave. The most overworked, stressed out parts go down first. In this case, the cell's own internal makeup is what really matters.
And look, this whole debate is built on more than a century of detective work. It goes all the way back to 1906, when Alois Alzheimer first saw those infamous plaques and tangles. And over the decades, we found more clues, identifying the key protein suspects one by one: tau, amyloid beta, alpha-synuclein. Each discovery has been another piece of this giant puzzle.
Okay, and this is where our story takes a really fascinating turn. We're going to zoom out from the brain for a second, out from the specific proteins and neurons, and connect this deep biological mystery to a concept that is also the driving force behind the revolution in artificial intelligence. And that concept is called emergence. So, what on earth is emergence? It's actually a pretty simple but totally profound idea. The motto is, more is different. It's the process where you get these incredibly complex, sophisticated, and often totally surprising behaviors that come from a huge group of simple little things, all just following a few simple rules. Think about a flock of birds, how they move together as one beautiful shape. There's no leader bird choreographing it all, it just emerges. This quote I found from a Reddit user just nails the connection perfectly. They suggest we think about artificial neural networks, or NNs, like a famous computer simulation called Conway's Game of Life. Each artificial neuron is like one simple little cell following a basic rule. But when you combine billions and billions of them, well, you get these mind-blowing new levels of emergence. And that's exactly what's behind the incredible power of today's AI models.
And when you see it like this, the parallel is just stunning. On the left, a real biological neuron. It's a wet, messy, living thing. On the right, a super simplified artificial neuron. It's basically just a math function. They're made of completely different stuff. But could the principle, the way they create complexity from simplicity, be the same?
And that right there is the key takeaway. In both our own brains and in artificial intelligence, the system is truly, deeply more than the sum of its parts. It doesn't matter if it's the emergent pattern of a disease spreading through the brain or the emergent intelligence of an AI that can write a poem. The magic isn't in the individual pieces, it's in how they all interact.
So where do we go from here? What does all this mean for the future? Well, this shared principle of emergence, it's opening up a whole new frontier, a new way of thinking about our biggest challenges in both neuroscience and in technology.
It leaves us with this really tantalizing question. As we get better and better at building and understanding these emergent AI systems, could those lessons give us a brand new way to look at the emergent chaos of a disease like Alzheimer's? And could studying how brains fail teach us how to build AI that's stronger and more resilient?
And that brings us to this final kind of wild thought. If the human brain and the most advanced AI are both just emergent systems running on different hardware, could we one day understand them so well that we could actually debug a failing brain the same way a programmer debugs a piece of software? It's a really profound idea, and it might just hold the key to finally solving one of the greatest mysteries in all of medicine.
Ever wondered how your brain manages to remember a face you saw years ago? Or, learns a completely new skill? Or even just feels the simple sensation of touch? It really all boils down to this tiny, intricate, and honestly pretty amazing communication happening between your brain cells. Today we're embarking on a really fascinating deep dive right into the electrical heart of our brains. We're going to explore the world of neurons, these rapid fire signals. They send action potentials, and how these fundamental electrical sparks literally wire themselves together to enable everything. Your memories, your ability to learn something new. Our sources, they range from places like Harvard Extension to some really cutting-edge scientific research papers. They'll be our guide through the, well, frankly, surprising facts that underpin our thoughts and actions. Our mission today is to pull out the most important nuggets of knowledge about how your brain communicates, how it adapts, and fundamentally how it makes you, you. Okay, so let's try and unpack this. Before a neuron can even send a signal, it's apparently in this highly active resting state. I'm trying to picture that. How exactly does the neuron manage to keep this delicate electrical balance just poised and ready to fire? It sounds like it's constantly working against something.
It absolutely is. It's a constant, very energy-intensive process. Think of it maybe like a finely tuned instrument held at just the right tension, ready to play a note. And at its core, a neuron has these main parts. You've got the dendrites, which are like antennae. receiving information. That info gets processed in the cell body. Then the axon, that's the long cable, carries the electrical signal, sometimes over surprising distances. And finally, the axon terminal passes that signal onto the next cell. Oh, and when you see a whole bundle of these axons traveling together, that's what we call a nerve. Right, and even in this so-called resting state, there's this incredibly precise electrical balance. A typical neuron sits at about minus 70 millivolts, the resting potential, which just means the inside of the cell is less positive or, well, more negative than fluid outside. It's definitely not passive. It's a dynamic equilibrium, constantly maintained.
Okay, so it's resting, but also constantly working. What are the key players keeping this electrical tension going? It sounds like a pretty sophisticated dance of charged particles.
Precisely. It really is a dance. The main players are ions. These are charged particles. Particularly, we're talking about positively charged sodium, Na+ ion, and potassium, K+ ions. At rest, they're spread out unevenly. You've got way more sodium outside the cell and much more potassium inside. This difference in concentration creates what we call a chemical gradient. But hold on, because these ions are charged, there's also an electrical gradient, right? More positive charge builds up outside the cell at rest. Put those two together, the chemical and electrical forces, and you get this powerful electrochemical gradient. It's always sort of pushing for things to even out. Now, to stop that from happening immediately, ions can't just wander across the cell membrane. They need help. They rely on specialized protein gateways ion channels. Some of these, we call them leak channels, are always a little bit open, letting a tiny trickle through, but others are gated. They only open when they get a specific signal, like a change in voltage, or maybe a chemical messenger binding to them. But the real powerhouse, the absolute workhorse maintaining this resting state, is the sodium-potassium pump.
The sodium-potassium pump. Okay.
Yeah, think of it like a tiny molecular bouncer. It actively kicks 3 sodium ions out of the cell for every 2 potassium ions it brings in. Critically, it's working against their natural gradients. And this takes energy, a lot of energy actually. It uses ATP. And get this constant pumping accounts for something like 20 to 40% of the brain's total energy use.
Wow, 20 to 40% just for keeping cells ready. That's incredible.
It really is. It's what keeps that neuron perfectly poised, like that coiled spring we mentioned,
Yeah.
always ready to fire.
Okay, that's a staggering amount of energy for something technically at rest. So, with all that delicate setup, what finally triggers the neuron? How does it actually spark and send its message?
Well, if the incoming signals, usually picked up by the dendrites, are strong enough to push the membrane potential from that resting negative 70 millivolts up to a certain point, a threshold voltage.
The threshold.
Exactly. It's typically around minus 55 millivolts. If the stimulation reaches that threshold, usually at a spot called the axon hillock, the trigger zone, then bam, it triggers an action potential, the neuron fires. And importantly, it's an all-or-nothing event. There's no half-firing. For a given neuron, once it crosses that threshold, the action potential happens fully, always with the same size, the same amplitude.
Okay, walk us through that moment then, the spark itself. What's the rapid-fire sequence? It sounds incredibly precise.
Oh, it is. It happens in very distinct phases very quickly. First up is depolarization. When that threshold is hit, special voltage-gated sodium channels snap open. Remember that electrochemical gradient pushing sodium inward? Well, now it rushes into the cell. This flood of positive charge makes the inside of the membrane rapidly less negative. It shoots past 0 and even overshoots to about positive 30 millivolts. That's the big rising phase of the signal.
Okay, so positive ions rush in, flips the charge.
Yeah.
Then what?
Then comes repolarization. Those sodium channels, they quickly slam shut, or rather inactivate, so no more sodium coming in. Almost at the same time, different channels, voltage-gated potassium channels, start to open, though a bit more slowly. Now, positive potassium ions rush out of the cell, following their gradient. This outward flow of positive charge makes the membrane potential swing back down, becoming negative again. And finally, there's often a brief hyperpolarization. Because those potassium channels are a little slow in the uptake closing, the membrane potential briefly dips even more negative than its resting state, like an undershoot.
An undershoot, okay.
Before it quickly levels back out to the familiar negative 70 millivolts. And remember, throughout this whole lightning-fast sequence, that sodium-potassium pump is working away in the background, restoring the original ion concentrations, getting everything ready for the next potential signal.
That is an astonishingly quick and complex sequence for just one spark. But thinking about it, what stops these signals from just bouncing back and forth, or firing uncontrollably, like a short circuit in the brain?
Excellent question. That's where refractory periods are absolutely essential, crucial for proper brain function. There are two main types. First, the absolute refractory period. This happens right during that repolarization phase, when the sodium channels are inactivated. During this tiny window, they cannot be triggered to open again, no matter how strong the incoming signal is. It's like the trigger mechanism is jammed for a moment.
Okay, so absolutely cannot fire again.
Exactly, and this is vital. It ensures, one, that the action potential travels in only one direction down the axon away from the cell body, a one-way street, and two, it puts a limit on how fast the neuron can fire, preventing, runaway uncontrolled activity. Then right after that, you have the relative refractory period. This lines up with the hyperpolarization phase, that undershoot where the cell is extra negative.
Right, when it dipped below the resting level.
Yes, precisely. During this time, the neuron can fire again, but because it's starting from a more negative point, further away from the threshold.
It needs a bigger push.
Exactly. It requires a stronger than usual stimulus to reach threshold and trigger another action potential. So these refractory periods together act like the brain's built-in control system, ensuring communication is precise, directional, and controlled.
Okay, so the action potential itself, the spark, is all or nothing. Its size doesn't change for a particular neuron, but clearly we experience things with different intensities, like intense pain versus just a gentle breeze. How does the neuron signal that difference if the spark size is fixed?
That's a really great point. You're right. The amplitude, the height of the action potential spike is fixed. But what can change dramatically is the frequency of firing. A more intense stimulus like that sharp pain will cause the neuron to fire action potentials much more rapidly, more spikes per second.
Ah, so it's the rate of firing.
Exactly. A gentle breeze might just trigger a few sporadic action potentials, a low frequency. The brain interprets this frequency, this rate of firing, as the intensity of the sensation or signal. And of course, how quickly these signals travel down the axon is also critical. We call this conduction velocity. Some axons transmit signals way faster than others. The main reason for this speed difference: myelin sheaths.
Myelin. I've heard of that. It's like insulation.
Precisely. It's a fatty layer of insulation wrapped around the axon, formed by special helper cells called glial cells, Schwann cells in the peripheral nerves, oligodendrocytes in the brain, and spinal cord. Think of it just like the plastic coating on an electrical wire. It prevents the electrical current from leaking out.
So our brains basically have these built-in, like, super speed highways alongside the regular roads. That's quite an engineering feat.
It really is. This myelin insulation allows for something called saltatory conduction. It's fascinating. The action potential doesn't smoothly travel down the whole axon membrane. Instead, it effectively jumps from one tiny, unmyelinated gap in the sheath to the next. These gaps are called nodes of Ranvier.
It jumps.
Here's the jump, yeah. This saltatory conduction massively increases the transmission speed. We're talking speeds over 100 meters per second in some myelinated fibers. And it's also more energy efficient. It's a brilliant evolutionary adaptation for rapid communication. And it's worth noting, these fundamental electrical signals, action potentials, aren't just for neurons in our brains. They also happen in other excitable cells.
Like where else?
Like muscle cells, for instance. And action potential is what triggers a muscle cell to contract. And even some plant cells use similar electrical signals, though they use different ions and mechanisms. It just highlights how fundamental this cellular spark is across biology.
It's truly remarkable to think that all our thoughts, our memories, the skills we learn, they're all built on these incredibly fast, tiny electrical signals. But how? How do these fleeting sparks actually combine to create the rich tapestry of learning and memory that defines us?
Well, that's the big question, isn't it? And what's really fascinating is how deeply learning and memory are linked. You could argue you've only truly learned something once it's stored in some kind of memory trace. Neuroscience often defines learning simply as the acquisition or maybe the strengthening of information in memory. And the substrate for this, it's that immense network, roughly 86 billion neurons in the human brain.
86 billion.
Connected by maybe 150 trillion synapses. It's this dynamic, interconnected network that gives us our incredible computational power.
We often just talk about memory like it's one single thing, don't we? But the research, like the famous case of Henry Molaison, or HM, shows it's much more complicated.
Oh, absolutely. Memory is definitely not monolithic. The case of Henry Molaison, HM, was absolutely pivotal. Back in 1953, he had surgery to remove parts of his brain, including the hippocampus on both sides, to treat severe epilepsy. The good news was, it did help his seizures. The devastating news was, it left him with profound anterograde amnesia. He couldn't form any new long-term memories after the surgery.
So he couldn't remember anything new that happened to him.
Pretty much. If you met him, chatted for a bit, left the room, and came back 5 minutes later, he would have no conscious recollection of ever meeting you. Yet, and this was the crucial part, he could remember things perfectly well from before his surgery, his childhood, world events. And even more strangely, perhaps, he could actually learn new skills. For example, he learned a task where he had to trace a shape while only looking at his hand in a mirror.
That's tricky.
Very tricky. And day after day, he got better at it, faster, more accurate. But every time he did it, he claimed he'd never done it before, had no memory of practicing.
Wow, so he could learn a new skill but not remember learning it. That sounds like a contradiction, almost. What did that teach us about different kinds of memory?
Exactly. That apparent contradiction was the breakthrough. It was powerful evidence that different types of memory exist and rely on different brain systems. We generally divide long-term memory into two broad categories now. First, there's declarative memory. This is stuff you can consciously recall and declare facts, events. And this is further split into semantic memory, that's for general knowledge, facts, word meanings, like knowing Paris is the capital of France, and episodic memory, that's your personal history, recalling specific events, like what you did on your last birthday. The other major category is procedural memory. This is for skills, habits, things you learn through practice, and often do automatically, unconsciously, like riding a bike, typing, and tying your shoes.
Okay, so H-on lost the ability to form new declarative memories, both semantic and episodic.
Correct. His hippocampus was essential for consolidating new declarative memories. But his procedural memory system was intact, allowing him to learn that mirror tracing task. Old memories were also stored elsewhere.
That really clarifies things. It also leads to this really mind-bending idea. How is specific knowledge actually physically coded in these huge networks? How does the brain represent, say, your grandmother?
Right. This brings us to the somewhat playfully named concept of the grandmother cell. It was a theoretical idea from the 60s. Could there be just one single neuron that fires only when you see, hear, or think about your grandmother?
Just one cell for grandma.
It sounds a bit simplistic, and it probably is. But the core idea that specific concepts are represented by the activity of specific neurons, or more likely specific groups of neurons, is very powerful. And actually, there was a more recent real-world discovery along these lines. Back in 2005, researchers working with epilepsy patients who had electrodes implanted in their brains found something remarkable. They found individual neurons that fired very selectively. For instance, one patient had neurons that fired strongly when shown pictures of the actress Jennifer Aniston, but not other famous people, places, or objects. They jokingly called them Jennifer Aniston cells.
Seriously, Jennifer Aniston cell.
Yep, but it's crucial to understand it's almost certainly not just one cell. That cell is likely part of a larger network. Knowledge, whether it's your grandmother or Jennifer Aniston, is probably coded by the unique pattern of activity across a whole assembly or network of connected neurons. It's the connections and the patterns that matter.
Okay, so concepts are coded in networks. How do these networks actually form? And how do they strengthen or weaken as we learn new things, have new experiences? How does the brain physically change?
This brings us right to the heart of it, synaptic plasticity. This is the truly remarkable ability of synapses, those connections between neurons, to change their strength and efficiency over time. This plasticity is the biological basis of learning and memory. It's summed up beautifully by that famous phrase, often attributed to psychologist Donald Hebb, neurons that fire together, wire together. It's called Hebbian learning.
Fire together, wire together. Okay.
The core idea is pretty intuitive. If neuron A repeatedly helps to fire neuron B, the connection, the synapse between them gets stronger. Think about it associatively. If you frequently experience the smell of lavender, say at the same time you visit your grandmother, well, the neurons representing lavender smell and the neurons representing grandmother are firing together. Over time, the connection between those sets of neurons strengthens. So in the future, maybe just smelling lavender is enough to trigger the grandmother network, bringing back memories. That's spreading activation across strengthened connections.
That makes sense. But what's actually happening down at the microscopic level? How does that synapse physically get stronger or weaker? It sounds like there's some tiny construction crew at work.
There absolutely is. It's molecular construction. On the strengthening side, the most studied mechanism is long-term potentiation, or LTP. LTP is a long-lasting strengthening of synaptic connections. It's considered a really prime candidate for how our brains rapidly store information, especially in areas like the hippocampus, which we know from HM is vital for new memory formation.
Okay, LTP for strengthening. How is it triggered?
It's typically triggered when there's a strong, high-frequency stimulation of the synapse, leading to a significant increase in calcium ions entering the postsynaptic neuron, the receiving neuron. Think of calcium as a key messenger activating internal processes. Often this calcium comes in through special receptor channels called NMDA receptors. This calcium surge basically tells the cell, hey, this connection is important, strengthen it. And one major way it does this is by inserting more receptor proteins, specifically AMPA receptors, into the postsynaptic membrane at that synapse.
More receptors means.
More receptors means the receiving neuron becomes more sensitive to the signal from the sending neuron. The same amount of signal now has a bigger effect. LTP can even unsilence synapses that weren't previously working by adding these AMPA receptors. And it's not just molecular. LTP is also linked to actual physical structural changes. Those little protrusions on dendrites where synapses often form, called dendritic spines, they can actually grow larger, change shape, even split to form more connections. It's making the connection more robust and lasting.
Wow, so the brain is literally rebuilding itself based on activity. What about the weakening side? Connections must weaken too, right?
Absolutely. That's where long-term depression, or LTD, comes in. As the name suggests, it's a long-lasting weakening of synaptic connections. Interestingly, while LTP is triggered by a large influx of calcium, LTD seems to be triggered by a more modest but prolonged increase in postsynaptic calcium. Different levels of the same messenger trigger opposite effects.
Okay, so less calcium means weaken the connection.
It seems so. This smaller calcium signal leads to the removal, or internalization, of those AMPA receptors from the synapse. Fewer receptors mean the synapse becomes less responsive. LTD is also associated with the shrinkage or even elimination of dendritic spines. It's really important to realize that both LTP and LTD are crucial. They work together constantly, dynamically sculpting our neural circuits, strengthening important connections, weakening less important ones, fine-tuning everything based on our experiences. It's how we learn, adapt, and also forget or refine information.
This ability to wire and rewire is just astonishing. But what happens when these really intricate processes, the signaling, the plasticity, go wrong? What are the consequences when this delicate system malfunctions?
Well, unfortunately, problems with these fundamental processes are implicated in many brain disorders. Structural and functional abnormalities in these neuronal networks, leading to altered patterns of brain connectivity and plasticity, seem to be key features in a lot of complex neurological and neuropsychiatric conditions.
Like, for instance, epilepsy. Our sources mention that. How does that fit in?
Yes, epilepsy, particularly types like temporal lobe epilepsy, TLE, is a prime example. Research clearly shows that seizures often originate in and spread through specific neuronal networks. The entorhinal cortex, which connects to the hippocampus, is often involved. A key issue in some forms of epilepsy can be a breakdown in the brain's inhibitory systems, specifically the GABAergic system, which normally acts like the brakes on neuronal activity. If these brakes are weakened, perhaps due to changes in receptors or neuron loss, you can get the excessive synchronized firing that characterizes a seizure. Plasticity can even go wrong, strengthening seizure pathways.
So understanding this plasticity, both normal and abnormal, must open doors for potential treatments, right? Knowing the mechanism seems like the first step to figuring out how to fix it.
Absolutely. It's a huge area of therapeutic research. For example, think about addiction. We now know that exposure to drugs of abuse can hijack the brain's natural plasticity mechanisms. Drugs can induce very strong, persistent LTP-like strengthening in synapses within the brain's reward pathways, like those involving dopamine neurons in the ventral tegmental area, VTA. This abnormal strengthening is thought to underlie things like craving and behavioral sensitization. Understanding how this happens is critical for developing treatments to reverse or counteract it.
Okay, addiction. What about other conditions?
Well, in Parkinson's disease, which primarily affects motor control, there's evidence pointing to problems with plasticity in a brain region called the dorsal striatum. Specifically, a form of LTD mediated by the brain's own cannabis-like molecules, the endocannabinoids, seems to be impaired. And intriguingly, some studies have shown that drugs designed to rescue or restore this specific type of LTD can actually reduce Parkinsonian motor symptoms in animal models. That points to a really valuable potential therapeutic target.
That's fascinating, targeting the specific plasticity mechanism that's gone wrong.
Exactly. And on a more fundamental level, all these intricate processes, building channels, pumps, receptors, changing structures, they all require the right building blocks and energy. That's why basic nutrients are so important. Vitamin B12, for example, in its active form, methylcobalamin, is absolutely crucial for the normal functioning of the entire brain and nervous system. There's even research looking into its potential analgesic effects for certain types of pain, possibly by influencing nerve function. It highlights how overall neuronal health supports these complex processes.
And thinking about it from the other side, not disorders, but just everyday life, these principles must have huge implications for how we should approach learning, right?
Oh, definitely. Understanding synaptic plasticity really informs how we can learn more effectively. Think about rote memorization, just repeating something over and over. It can work, but it's often difficult and boring, right?
Yeah.
That's partly because it doesn't always strongly engage these associative fire together, wire together mechanisms. In contrast, associative learning, actively linking new information to things you already know, is much more powerful. You're literally building on existing strengthened networks. And active recall, things like testing yourself, explaining a concept to someone else, even just trying to remember information without looking it up, these are incredibly potent. Why? Because retrieving the information forces those neural connections to fire again, strengthening them further through LTP-like processes. It consolidates the learning.
So things like using mnemonics or flashcards where you have to recall the answer.
Exactly. Mnemonics are a classic example of creating strong, often unusual associations to make information stick. Active recall activities directly reinforce the pathways you want to strengthen. It's leveraging the brain's natural rewiring toolkit.
Wow. Okay. Today we have gone on a real deep dive. I mean, from that instantaneous electrical spark of an action potential, all the way to this incredibly complex, subtle dance of synaptic plasticity that really underpins our ability to learn, to remember, to adapt.
Yeah, we've traced how simple movements of ions create these complex signals, how myelin helps them travel super fast, and, crucially, how the very structure of our brain connections literally changes with our experiences with every new thing we learn. And we've also touched on how disruptions in these processes can lead to significant neurological problems.
It's just a profound reminder, isn't it, of how incredibly dynamic and adaptable our brains really are. It's constantly changing. So thinking about all this, what really stands out to you about this incredible machinery inside our heads? Maybe consider this. If learning is fundamentally about strengthening and creating connections between neurons, and if actively recalling information further solidifies those connections, how might just consciously taking a moment to reflect on what you've just learned, maybe even trying to explain it simply to someone else, be one of the most powerful learning tools you have? You'd be actively, consciously rewiring your own brain in real time.
That's a fantastic thought to end on. Keep exploring, keep connecting those ideas, and definitely keep asking questions.